Hello, class. This is Professor Fisher, continuing on with the nervous introduction to the nervous system. One of the systems that's really helps control our body. Now we're going to get into some of the basics of nervous tissue. Remember, we talked about a couple of different tissue types in chapter 4. We talked about epithelial tissue, covers. We talked about connective tissue, connects the body, supports structure, helps give the body some protection, structure in different ways. Skeletal, which also helps with structure and protection. Skeletal is part of the connective tissue. It helps with structure and protection. We talked about muscle tissue, which generates a force. And we talked about nervous tissue. All right, so our 4 tissue types. And so now we're getting into more detail with nervous tissue. Had a little brain fart there. Pardon me. 4 types of tissue: epithelial, connective, nervous, muscle. All right, so let's go into, remember, tissues are made up of different cell types working together for a specific function. That is what definition of a tissue. So what are the cells that are working in nervous tissue? There's neuroglia. Okay. Neurons are excitable cells responsible for sending and receiving signals, which we call action potentials. We're going to talk all about those, about action potentials and how they work and how the signal is actually sent and received. So we're going to get into a lot of detail as far as that's concerned. That's a pretty intense process, but very important, because that's basically the purpose of nervous tissue, to receive signals, as it translates our interaction with our environment, and also internally, and also to send signals to tell our muscles, tell our heart, tell our different organs to do different things. All right, so this is very important. But what is the structure of this cell, nerve cell, that can send and receive signals? Right, it pretty much has 3 major parts. It has a cell body, which is, the cell body is what we think of when it's called the soma, or the soma cell body, or the cell body for a neuron. That's basically the part of the cell that is very similar to every other cell. Right, so this is the most metabolically active region. Manufactures all proteins needed for the whole neuron. That's where the organelles are that are in the neuron. Right, so in the cell body for this neuron, you have the free ribosomes, rough endoplasmic reticulum. You have the Golgi apparatus. You have the nucleoli, the ribosomal RNA. So all of these things that you would find in a normal cell. Mitochondria supply energy. It needs energy just like nerve cells need energy, just like any other cell. You also have cytoskeleton, structural support, but you also have these neurofibrils, intermediate filaments of the cytoskeleton, which extend into the neuron processes, which we'll talk about. All right, so very similar to any other cell as far as the cell body is concerned. Then we have the dendrites, right? Dendrites are specific to nerve cells, short branched processes, and they receive input. That's the key term here. Remember, if you remember anything about the dendrites, they receive input from other neurons. So you'll see that a neuron a lot of times has multiple dendrites usually, but there are cases where it's not multiple. But most neurons have multiple dendrites, and that's where they receive information from other cells. Right, so these are short, like branched processes, right? And they transmit the signal towards the cell body. Okay. And then each neuron has only one axon. So you can have multiple dendrites, you can receive multiple signals, but you only have one axon. Axon can generate and conduct an action potential. That is going to be a very key term in for the next couple chapters, action potentials. Okay, so remember that term. It's conducted in the axon. That's your nerve fiber. This is what we generally think of when we think of nerves. When we're talking about nerves, we're really talking a lot of times specifically about the axons, because the axons are the part of the nerve cell. Remember, we're still talking about the nerve cell. It's a part of the nerve cell that generates the action potential, so it sends the signal, right? And we break down the axon into different segments. Axon hillock, and that's where it originates. That's the closest section to the cell body. Then you have axon collaterals, where it can branch, extending from the main axon. Telodendria, small branches rising from the axon, and axon collaterals, near where extensions end. At the ends, you have the axon terminals, components that communicate with the target cell. We'll talk about those in more detail. And then you have the axal lemma, which is the plasma membrane surrounding the axon and its cytoplasm. All right. This is a microscopic view of a neuron, neuroglia, extracellular matrix, dendrites, and axons. It's kind of hard to tell if you don't know what you're looking at here, but we have other diagrams that look a little bit better, like this one. So you see here, and it's important to understand. So you see here we have the soma cell body. So this, if you don't think about all the dendrites and the axons, this cell body looks very similar to chapter 3 cell, the standard cell. All right. Nerve cell looks like a standard cell, right? So that's basically your standard cell. You have your nucleus, you have your mitochondria, all the things you would find in a regular cell, right? But for a nerve cell, you have these extensions called dendrites. These are going to receive signals. That's ways that other cells communicate with the nerve cell, and they can send the signal via this axon. So this axon, you could have an axon collateral. So the signal could go down both of these sort of pathways. The axon hillock is the section right next to the cell. Axoplasm is the cytoplasm of the axon. Telodendria is going to be the sort of branch sections here that are going to eventually lead to the communication with the target cell. The axon terminals, that's the end of the axon. That's the actual end that actually makes, it's going to basically help, the last part of the axon that's going to basically make contact with the target cell. And this target cell could be anything. It could be an organ, it could be muscle, it could be, anything in the body, depending on what where this nerve is and what it's communicating. Okay. Notice that one neuron, as we said earlier, can have multiple dendrites, but only one axon. Even though the axon can have collaterals, branch some, right? So it can receive information from multiple places, but it sends information only through one pathway. All right, so this is further describing that neurons have 3 main functional regions. Okay? So receptive region, dendrites in the cell body, that's where we receive our information. Conducting region, That's your axon. We're sending our signal. So we received whatever signal we've received. Now we're sending the signal that we're going to send now. That's our conducting region. And then secretory region. Secretory region at the end, where we secrete the signal. Axon terminal. Right, you have different types of neurons. Multipolar, single axon, multiple dendrites. That's 99% of your neurons. Right, that's basically what we've seen so far. Single axon, multiple dendrites. That's if you see a neuron in the body, that's probably what it's going to be. But you also do have other types. You have bipolar, one axon, one dendrite. You'll see this in the eye, for instance. You're only receiving, one kind of input, and you're sending that information, basically to the brain, eventually to the brain. The eye and the nose, you'll see this type of activity where they're receiving one thing. The eye is simply receiving light, and you're converting that into a signal that you're sending that information to the brain, which your brain is going to integrate and convert and tell you what it is. And then you have pseudo unipolar. Only one fused axon extends from cell body, divides into two processes. And so we see this in when we have pain, touch, and pressure from cell body to spinal cord. Okay. This is going to come up in a picture a little bit in another slide. So here we go. We have sensory, your afferent neurons carry information. These are our functional groups. These are our functional groups for our neurons. We already talked about these a little bit, but when we talked about the peripheral nervous system, we generally have sensory afferent neurons carry information towards the CNS, right? So your neuron cell bodies in the PNS receive information from sensory receptors, relay the information via axons to the brain and the spinal cord. Those are usually those pseudo unipolar or bipolar, right? Sensory neurons, right? So for instance, the neurons in your eye, they're sensing the light that's reflected, that's reflects into your eye. They're sending that information to the CNS. It's actually a cranial nerve, so it goes straight to the brain. That's usually that's actually a bipolar nerve. You have interneurons, which relay information within the CNS. So once something gets into my brain, and we're going to talk about this even in more detail. So I see something, the neuron sends the signal to my brain. Once it gets in my brain, now multiple neurons are firing in my brain as I'm processing what I've seen. Okay, those are usually multipolar neurons, communicate with many other neurons. Multiple parts of my brain are working. What is this information that has been sent into my brain? And then we have motor neurons carry information away from the CNS. Remember, the CNS is your brain and your spinal cord. So now that I've processed this information, I've seen I've seen this slide, I'm done with this slide. It sends that information to my brain. Now my brain tells my finger to move, the muscles in my arm and my finger to move, to press the next key on my computer to go to the next slide, right? So I know that's a quick example, but that's how the body works, and it's amazing. Right, so these are just showing you the different classes of neurons, okay? Multipolar neurons, axon, multiple dendrites, bipolar neuron, one axon, one dendrite, pseudopolar neurons, all right? Peripheral, axon, central axon, right? No dendrites. All right, typical, this is motor, most neurons in the CNS, motor neurons in the PNS. This is your special senses. We have a whole chapter on special senses, your eyes, your nose, and these are your sensory neurons. Now, specific neuron components, we do group them together. All right, so in your CNS, we have what we call nuclei, clusters of neuron cell bodies, and tracks, we call tracks bundles of axons. Don't get confused, but in the PNS, your peripheral nervous system, we use a little bit different terminology. And so this is where it can get a little confusing. Clusters of neuron cell bodies in the PNS are called ganglia. Clusters of nerves, clusters of your bundles of axons, like I said earlier, what we generally call nerves, I don't mean when you say like somebody's getting on my nerves. I mean like when we say like somebody has nerve damage or something like that, what we're generally referring to as nerves are bundles of axons in your PNS, in your peripheral nervous system. So nerve cells, their axons are extending out, and you have multiple neurons, multiple axons from multiple neurons bundled together, traveling together. When that gets like somehow hurt or injured, that's what we're generally talking about when we're talking about nerve damage. Okay. Right, but this is the medical terminology. So when we talk about nerves in the PNS, the term actually refers to bundles of axons. And the cell bodies, the soma cell bodies of neurons in the PNS is called ganglia. And then it's in the CNS, they have different names. Bundles of axons are called tracks, and the clusters of neuron cell bodies are called nuclei in the CNS. All right, now let's get into neuroglia. Neuroglia, we know, does not send a signal, but it does provide structural support and protection for neurons and helps maintain their environment. We're not going to spend a ton of time on the neuroglia in the next couple chapters, but it is important because they do play a special role. They're able to divide, fill space left behind when a neuron dies. And we're going to talk about, it's important to discuss what happens when a nerve is injured, what happens when a neuron dies. It's not like, it's a little bit different. Somebody out there may have had permanent nerve damage. What does that mean? You've like permanently lost feeling in part of your body, or what about paralysis? Permanently paralyzed. So all of that those are things that we need to, or we're going to address in the next coming chapters. All right, so neuroglia can fill the space when neuron dies. And there's different types of neuroglia, different types of cells that make up the neuroglia, and they're specialized for function. All right, so 4 types of neuroglia reside in the CNS and 2 types in the PNS. And the neuroglia is going to also it's going to help with the oligodendrocytes and the Schwann cells are going to really help with the signal sending of the axons and the dendrites specifically the axons in the neurons, which we'll see soon. All right, so let's talk about these different types of neuroglia. right? So, like I said, there's 4 types in the CNS, 2 types in the PNS. All right, so neuroglia. So we have astrocytes, large star-shaped cells, many processes terminating in feet. What is their function? That's basically what we want to know. They anchor neurons and blood vessels in place, transport nutrients and gases between blood vessels and neurons, help form this blood-brain barrier. You've probably heard of that if you know of anybody that suffered an injury to their brain. So they form this blood-brain barrier, which is a protective structure. Surrounds the capillary endothelial cells, makes them impenetrable to most polar compounds and proteins. They repair damaged brain tissue by rapid cell division, all right? So they help repair damaged brain tissue, astrocytes. Oligodendrocytes, you're gonna see these very important. Also in the CNS, radiating processes or flattened sacs, they wrap around axons of nearby neurons to form what we call myelin. Myelin is very important because that's going to help the signal, the signal of the axon move faster. And we'll talk about that whole process. So remember, oligodendrocytes help myelinate the axons in the CNS. Microglia, when you see the term micro, always think small. So of the neuroglia, the microglia, small neuroglia cells, activated by injury into wandering phagocytic cells within CNS, ingest disease-causing microorganisms, dead neurons, and cellular debris. So this is sort of your immune cell of the CNS, right? We have many, many layers of immunity when it comes to the body. So many layers of immunity. Our immune systems are amazing, but we have these microglia cells, which help remove disease-causing microorganisms that could get into your CNS, dead neurons, cellular debris. Right, and then we have this. Then we have ependymal cells. These ciliated cells manufacture cerebrospinal fluid, which is also going to be very important for the protection of your brain and spine. You do not find these cells in your peripheral nervous system, only in your central nervous system. Cerebrospinal fluid is only in where you have your cerebrum, your brain, and your spine. We'll talk about the importance of that a little bit later as well. All right, so these are your neuroglia for your central nervous system, your CNS. Then we also have 2 other types of cells which are located, 2 other types of neuroglial cells which are located in your peripheral nervous system. Schwann cells, these are the are similar to oligodendrocytes. They encircle the axons of the PNS and provide them with myelination. So very similar to the oligodendrocytes of the CNS, except these are located in the PNS and are a little bit different. And then we have these satellite cells, surround the cell bodies of neurons in the PNS and provide supportive functions, right? So they're kind of like the astrocytes of the PNS. All right, Schwann cells, satellite cells. All right, now let's talk about this myelin sheath. Very, very, very important Um, the myelin sheath is layers of plasma membrane, Schwann cells, or oligodendrocytes, depending on if you're talking about the PNS or the CNS. They wrap around these layers of plasma membrane, these Schwann cells or oligodendrocytes wrap around multiple layers neuroglial cells wrap multiple layers of membrane around the axon. So they literally wrap around the axon, sort of like insulation. If you ever look at a wire, like, you know, if you have something plugged in, your TV wire, well, don't cut it. But if you did cut your wire, you notice it has like the rubber on the outside, and then the actual wire, what actually is transmitting the electricity is like a metal. I don't know what it is, what kind of metal it is. It's like copper or whatever. I don't know what they're made of, what kind of metal the wires are, but that's the actual part that transmits the electricity. But the reason they cover it with the rubber or whatever the insulation is, the black rubber or something, is because if something else was to touch it, it would shock them, and that would be bad. But then also it would the electric would go off to the side, so to speak. Similar idea in the body, right? So the lipid content, remember, plasma membranes, phospholipids, the lipid content of myelin sheath insulates the axon. So you have this plasma membrane wrapped the extra plasma membrane. That's what these Schwann cells or oligodendrocytes do, wrap around the axon, prevents ion movements like rubber around the copper wire, increases the speed of the action potential. Myelinated axons conduct action potentials 15 to 20 times faster. Remember, action potentials is that ability to send the signal. So they're 15 to 20 times faster. That axon can send that signal 15 to 20 times faster than unmyelinated axons. You ever heard the saying, the hand is quicker than the eye? I don't know if that necessarily is true. I think Bruce Lee used to say that. I don't know who else said that, probably somebody else. But the body can be amazingly quick, especially when it's trained. It can be amazingly quick because it can send signals so fast. And you think, you know, the signal goes, you know, up my spinal cord to my brain, process back down to the muscles in my hand or whatever. Like I see something, sends a signal to my brain, then sends a signal to my spinal cord to send a signal to my arm to move or whatever. We used to play this game where you have, like, you put your hands on top of somebody else's hands, like your hands is on top of their hands, and you have to like see if you could flip your hand over and slap their hand. I don't know if anybody else played that game. I don't even know what it was called, but it was like, see how quick you were if you could slap their hand. If you missed like a certain number of times, if you missed, then it was your turn to go on top, and they had to try to slap your hands. So it was but it was just a test to see how quick you were. But it's so interesting just how fast the mind can work. But this is one of the reasons why, you know, signals can be received and sent so quickly. Like, you can literally, you know, you could see somebody's hand move in like a split second, move your hand away so that they couldn't slap it. Like, to be able to see that, process it, send the signal, move the hand in a split second. Amazing. But it's because your axons are myelinated, and therefore able to send signals. It allows your axons to be able to send signals so fast. Right, so differences in myelination between peripheral nervous system and central nervous system. Sorry, I get a little excited when I'm talking about the nervous system. It's pretty amazing. All right, so we have the neural lemma on the outer surface of myelinated axons and the PNS. Schwann cell nucleus, which also contains organelles and cytoplasm, not present in the CNS. Remember, Schwann cells are only in the PNS, not in the CNS. Number of axons myelinated, oligodendrocytes, which is the CNS, have multiple processes that myelinate multiple axons in the CNS. Schwann cell only myelinates one axon in the PNS. So these are just slight differences. So oligodendrocytes, one oligodendrocyte can myelinate multiple axons in the CNS, whereas a Schwann cell in the PNS only myelinates one axon. And the Schwann cell wraps itself around a axon in the PNS. And so you have the Schwann cell, the nucleus, which contains the nucleus, the cytoplasm, the organelles, all of that is present. Right? So this is what it looks like in just microscopic. Here we have the myelin sheath and the PNS and CNS. Right, that's what it looks like. So you have dendrites, soma cell body. This is your axon, and these are Schwann cells myelinating it with slight gaps in between called the nodes of Ranvier. Some people say Ranvier, but Ranvier. Right, you see that Schwann cell, the nucleus. So it's a normal cell, but it wraps itself around the axon. Take a cell and just literally wrap itself around. In the CNS, we have oligodendrocytes. See the cell body of oligodendrocytes, axons of a neuron, and of multiple neurons. And the oligodendrocyte can take and wrap extensions of its plasma membrane around multiple axons. Right? You get the same result, but they do it a little bit differently. All right, so you have these both in the CNS and PNS. Axons in both CNS and PNS are generally longer than neuroglial cells, so multiple cells must provide complete myelin sheath, which makes sense. One cell can't do the whole thing. One oligodendrocyte, one Schwann cell can't cover a whole axon. So it makes sense that you have multiple oligodendrocytes and Schwann cells that help cover the axons of the neuron. Right? But you have these internodes, segments of axon covered by neuroglia. All right, internodes. That's the section that's actually covered with the myelin sheath. And then we have these nodes of Ranvier, gap between adjacent neuroglia where myelin sheath is absent. And these are going to play an important role. All right, so you have these gaps in between, as you see here. So these little gaps. So the inner node is the part that's actually covered, and the gap is the node of Ranvier. Right? Now, this is something you probably have heard of, but maybe didn't know exactly what it meant. Right? So small axons in the CNS and PNS are usually unmyelinated, small axons. White matter, and we say that we use this term sort of freely, white and gray. We talk about the brain, and we're going to talk about this in a lot of detail. But white matter is composed of myelinated axons. They appear white. That's interesting, right? Somebody had an aha moment, hopefully, just right there. Gray matter, composed of neuron cell bodies, unmyelinated dendrites, and axons. So unmyelinated axons appear gray. So when they say gray matter, and we're going to talk about the brain, the spinal cord, the part that's myelinated is white. The part that's unmyelinated, cell body, they're always unmyelinated. Dendrites, unmyelinated. And axons, small axons, are unmyelinated. That's what's gray. Now we have to talk about regeneration of nervous tissue. We talked about this a little bit. I mentioned this a little bit briefly, and we're going to go into detail about this even more so, but this is important. Especially as this relates to injury. Regeneration or replacement of damaged tissue is nearly non-existent in the CNS. And I know that's a difficult thing to swallow sometimes. I remember my son they thought he was going to have surgery because his skull, he had some issues with his skull when he was born, and they were going to have surgery. They said that they thought they were going to have surgery on his skull because it wasn't, they thought it was going to put pressure on his brain. I think because it wasn't like expanding quickly enough or something like that. It was something along those lines. When they tell you when they say your son in surgery, your baby in surgery, you go. It's not a good feeling. I'll just put it like that, because I didn't know necessarily exactly what they were going to have to do, but I knew that if anything got injured in his brain, your brain basically usually doesn't heal itself. And so that brain damage could be a very bad thing. Let's put it like that. He ended up not having to have surgery. We ended up waiting it out. Things got better on their own. We did not have to have surgery on his skull or his brain. And so we were very happy. But I remember my wife crying when they told us the news initially, that they thought if the way things were going, that we were going to have surgery. And it wasn't, don't get me wrong, if my kid ended up mentally delayed or something, I would have still loved them just the same. It's just, you just get sad because, it's going to be a lot tougher life for your child, if they don't have full use of their brain. And it just, as a parent, they're going to struggle. But he's just fine. He's 10 years old now. And so he's just fine. Brain works just fine. He has a good sized head like his dad. All right. But so regeneration or replacement of damaged tissue is nearly non-existent in the CNS. So usually if that's why your brain is so protected. We already talked about cerebral spinal fluid, blood-brain barrier. We haven't gotten into the skeletal system yet, but the skull, very strong structure that protects the brain. Is also, we're going to talk about the different layers of the meninges and everything like that to help protect the brain. So many things that are protecting your brain right now, because if it does get damaged, it's usually permanent. There is limited regeneration in the PNS, peripheral nervous system. And that is, if the axon is damaged, you can replace that as long as the cell body of the nerve is not damaged. Okay. Another huge issue, obviously, are tumors. And my heart goes out to anybody that has experienced this or knows somebody close to you has experienced something like this, because another thing that can be incredibly detrimental is tumors in the brain. Originated primary brain tumors, mostly gliomas. So they're not really caused by the neuron, but it's caused by the neuroglia and division of the glial cells. It can be some things that can predispose you to brain tumors, or radiation, certain diseases. The most common neuroglia cells that becomes cancerous, forms a tumor, is the astrocyte. And if you look at this diagram, you see this tumor, you see this bulging in the head, these cells, and that's going to be extremely problematic to remove. And They have to go in, cut you open all the way, through your skull, and remove the mass, and then you got to do chemotherapy. So it's very detrimental, especially considering how important the brain is. And so that's just some of the issues you can have concerning the brain.